I'm Sherry Barkin. I'm a professor of pediatrics at the Vanderbilt University Medical Center. Welcome to an introduction to life course research and complexity. Life course research embraces complexity science, but complex is not complicated. Complicated means many parts, all different, and each with their own very precise role in some sort of machinery that creates a specific outcome. While complex means a system that's made up of multiple interacting parts that produces patterns and outcomes together. So as Aristotle said, the whole is indeed greater than the sum of its parts. In this talk, I'll review examples of using a life course lens to examine the emergence of health or disease across the lifespan. I'll describe complexity science and provide some examples of how this can change the way we ask and answer important questions to understand health and disease throughout one's life. So health and disease emerge over time. I'd like to tell you 3 stories to illustrate this. The first is the story of cytomegalovirus exposure and immune functioning. In early life, acquisition of CMV can lead to enhanced responsiveness to vaccines, whereas in later life, exposure to the same virus leads to decreased vaccine responsiveness. The timing of the CMV exposure has a profound impact on immune system priming and functioning. But this isn't the whole story. There are heritable and non-heritable influences, such as nutrition and one's microbiome, that also contributing to functioning and the development of immunity over a lifetime. The second story I'd like to exemplify is the development of Alzheimer's disease. I'm a pediatrician. You might wonder why I'm interested in Alzheimer's disease. And I think looking through the lens of what we can typically consider as an older person's disease, we realize that actually Alzheimer's is something that develops over a lifetime with many contributing factors. Genes play a role, but by themselves rarely predict if and when Alzheimer's will emerge. Epigenetic markers acquired from gestation to older age could modify gene expression, and that could induce or accelerate Alzheimer's. Moreover, non-heritable factors such as infections, stress, nutrition, adiposity, and exposure to toxins can have differing effects based on the degree and timing of the exposure, as well as the interaction of these factors. And the third story to highlight life course complexity is type 2 diabetes. So often when we think of type 2 diabetes, if you look just prior to the diagnosis, you probably have missed how type 2 diabetes has emerged over time and missed the opportunity for intervention and early prevention. Here, this is a story of early life exposures and the developmental origins of health and disease that posits that early life exposures can confer lifelong effects, either bending towards health or disease, and that this early life period is exceptionally sensitive to developmental plasticity. So type 2 diabetes, it's associated with many things: higher maternal pre-pregnancy BMI, hyperglycemia, maternal hyperglycemia, exposures to endocrine disrupting chemicals in gestation or early in that infant's life in a mother who's breastfeeding, the infant gut microbiome, which is being influenced by pre, peri, and postnatal factors. They all affect the risk of developing type 2 diabetes. But so too does exposure to poverty and adverse childhood experiences and engagement in risky behaviors in adolescence, like smoking and alcohol use. Exposure to any of these elements, from physiologic to sociologic, needs to consider dose, that's the amount of the endogenous or the exogenous exposure, duration, that's the short or the long-term accumulation of physical and psychosocial exposures throughout the life course, and timing, the timing of the exposure in relation to human development, with some periods allowing for more plasticity and adaptation. A conceptual way to bring all of these interacting factors together and understand the emergence of health or disease phenotypes is reflected in this Prism model introduced by Dr. Liz Goodman and colleagues. It represents an individual's health development, and it considers 4 dimensions of health, including experience, which is the impact of environment from molecular to systems level on the individual, performance, what the individual is able to do, how they function, adaptability, which is the individual's capacity to respond to internal and external changes and return to baseline or achieve a new baseline, and potential, which is what the individual may be able to accomplish if challenged at this moment or in the future. This occurs in multiple levels, and it occurs over time. Through this prism, you have the emergence of health or disease, and this allows, over the course of the lifetime, an understanding of different trajectories of health and disease. The common approach to handling these complex issues is to simplify. After all, traditional science is typically reductionistic. It's based on the Newtonian paradigm, looking for cause and effect. And this is a very powerful way to ask and answer some questions, but not all. The problem with simplifying complexity is that it ignores the interdependence of the factors over time, as those 3 stories illustrated. Often when we simplify complexity, we might look at independent and additive effects, or clustering of risk, or maybe a pathway model, or some sort of chain of risk. Unfortunately, this really looks at health or disease in isolation. And that's not actually how humans function or even nature functions. So take this flock of birds. A flock of birds is something that moves in time. But if I were to just look at one bird, I wouldn't really understand how these patterns emerge. Instead, I have to look at it all together over time. This is the definition of complexity science. Complexity science is a system where unexpected patterns emerge as a result of many individual parts interacting. And these interacting parts follow a set of rules that result in the emergent properties. That allows us to ask questions like, how does each individual part behave? And how do the parts of the system act together to affect the function of the whole system over time? By applying complexity science and a Lifecourse lens, we can ask and answer different questions. For example, why have we seen an increase in neurodevelopmental disorders like autism and schizophrenia? In a study led by Estes and authors, they examined a model of how maternal immune activation during pregnancy increases CNS disorders in the offspring, including autism and mental health disorders. This model posits that infection during gestation releases pro-inflammatory cytokines, and Th17 cells are then activated. This affects the developing fetal brain, along with specific exposures, such as childhood stress and nutrition, and/or adolescent drug abuse. All of these things then interact with a genetic predisposition, and this increases the psychiatric disorders that we see in adults. This application of complexity Lifecourse science requires new methodologies, access to large databases that can be integrated and harmonized and pooled over time, integrating data that includes genetics, environment, timing of exposures, and measurable health and/or disease outcomes. So what does this methodology look like? Here's an example carrying forth that question of maternal inflammation and how that might affect the developing brain in children and later outcomes. So in a study here conducted by Rudolph et al., the authors linked maternal inflammation during pregnancy to her newborn's functional brain architecture and then working memory 2 years later. This was done in a longitudinal cohort. They measured interleukin-6 in maternal blood during all 3 trimesters, and then applied functional MRI to characterize the brain architecture of the infant at 4 weeks of age. And then they used big computing power to design a model that they could test thousands of times. This allowed them to generate a model that they could apply to predicting working memory in the toddler who is then age 2. You can see that this is challenging and in fact seems complicated, but is rather instead complex. Because now we have new tools that we've never had before. We have much more computing powers. We have the ability to generate and utilize large data sets and integrate them well to identify phenotypes of health and disease. So a Lifecourse informed model applies complexity science and not only introduces a new way of thinking about how disease develops, but leads us to new approaches to treatment and prevention. How does this help us to chart the way forward for Lifecourse research and researchers? Well, we are positing that we need to move forward together to improve processes and improve applications. To improve processes, we need to integrate data sets across the lifespan. We need to design data science cores that can focus on curation, data cleaning, analysis, and data modeling. And we need new ways to model high-dimensional data. As we move forward to improve application, we need to expand traditional epidemiologic methods to include systems and network modeling and improve reproducibility and standardized measures. So our take-home message is that Lifecourse research embraces complexity science and doesn't simplify complexity. It allows for scientists to generate and test new hypotheses, and it requires integrated data sets to capture dynamic interactions over time. It's complex, but it's not complicated.